Good day, students. So we will now have the last PPT on medical entomology. And this time we will be talking about flies. So this is just a review of what we have already covered so far. We have discussed the crustaceans, chilopoda, diplopoda, arachnids, and in terms of the insects, we have finished discussing fleas, lice, mosquitoes, and cockroaches. So for this entomology 5 PPT, we will be discussing about flies. Before we'll go to the flies, let me discuss this particular slide with you guys. Arthropod is divided into what? Crustacea, Chilopoda, Diplopoda, Arachnid, and the insects. I want you to take note that the insect group is further subdivided into this one. You have the Malophaga, which is best represented by the chewing lice. As I know I have already discussed chewing lice in, I think, entomology part 3. Okay, I told you that they have the same flattened bodies as the sucking lice that can infest humans. Okay, what's make them different from the sucking lice is that they have bigger heads. Okay, and then they particularly target birds. Okay, then you have the Anoplura, best represented by our sucking lice, the Tyrus pubis, the Pediculus humanus corporis, Pediculus humanus capitis. Okay, and then you have Hemiptera. Okay, best represented by blood-sucking bugs. I think the one that is medically important is the triatomine bug, the kissing bug. Okay, and also bed bugs. Okay, then you have the Diptera represented by flies and mosquitoes, and the Siphonaptera, which is best represented by the fleas. So please spend some time memorizing the subgroups of insects and the examples. Malophaga group is best represented by the biting lice. Just like the sucking lice that infest humans, they are wingless, they have flattened bodies. What sets them different from the sucking lice is that they have bigger heads and they particularly target birds or any animal with feather. So these are two biting lice. As you can see, they have flattened bodies similar to Pediculus humanus corporis, Pediculus humanus capitis. It's just that they have bigger heads. And as you can see in the picture, they particularly target avian host. Anoplura is best represented by the sucking lice, the ones that we have studied on entomology part 3. Okay, you have the Pediculus humanus corporis, Pediculus humanus capitis, and the pubic lice, the Tyrus pubis. The next group of insect, the Diptera, which is represented by mosquitoes and flies. Okay, so we will now discuss the flies. Then we will discuss the Hemiptera on the last part of our PPT. Okay, so if you can remember, Malophaga, Anoplura, Hemiptera, okay, Diptera, and Siphonaptera. I think we have already finished discussing the fleas, the Cynoptera. So we will first start with Diptera, then we'll go to Hemiptera later on. Okay, so for the flies, one of the medically important flies is the housefly, okay, the Musca domestica, okay, and you have learned from our entomology 3 PPT that they have mouth parts designed for what? Sponging and lapping. And you know very well, houseflies are mechanical vectors of amoebiasis, hepatitis A and E, typhoid fever, cholera, what else? Dysentery, okay, any disease that can be transmitted through fecal oral route. But there are flies that can bite, okay, and we have already studied them in our previous parasitology topics. You have the Glossina fly, which can transmit Trypanosoma brucei. We have the Phlebotomus fly that can transmit Leishmania species. Then you have the Chrysops fly, the deer or the mango fly. I was about to say the dango fly. The deer or mango fly, the one that transmits Loa loa. Then we have the Simulium fly or the black fly, which can transmit Onchocerca volvulus. So let's start with this biting fly. Can somebody please identify this one? Okay, because of its yellow color, this one is also known as the mango fly. Since it particularly targets deer, this one is called the deer fly. Ladies and gentlemen, this is the Chrysops fly. And I hope you can remember that the Chrysops fly, the deer fly, and the mango fly is the vector, insect vector of what? Loa loa. Okay? And I want you to take a look at the life cycle of Loa loa. the mango fly or the chrysops fly will introduce to the human host the stage 3 larva of the parasite, right? Okay. And I want you to take note at number 4, part of the life cycle, what is taken by the mango fly or chrysops fly during the blood meal is the microfilaria. The microfilaria will progress to form the stage 1, the stage 2, and the stage 3 larvae. As you can see, there's no further increase in the number of the parasite. The parasite just undergone transformation in terms of the stages. So therefore, this chrysops fly is an example of what type of biological vector? Propagative, developmental, or cyclopropagative? So I'll leave that to you to answer, ha? Okay. You have learned from the discussion on the loa loa that the microfilaria will go to the blood. Okay, that's why we can use the blood smear as the diagnostic specimen for diagnosing loa loa. The problem is loa loa adult tends to migrate in our body. That's why at some point it can cause the calabar swelling, the allergic reaction to the migrating adult, and at some point it can even reach the eye. And for the patient to be treated, the worm has to be manually or surgically removed. But this loa loa is not known to cause blindness. In terms of the chrysops fly, only the female fly bites. Okay? And I want you to take a look at the mouth parts of a chrysops fly. Can you appreciate that the mouth parts basically look like that of scissors? Okay? So they have mandibles and maxilla that can actually slice through the skin so that the organism can feed on the blood. Okay? And just like in the case of mosquito, they can also introduce anticoagulant through their saliva. And the problem is the saliva might contain the stage 3 larva of loa loa. So this one is just to show us the mouth parts of a loa loa fly. Can you appreciate the scissor-like mandibles and maxilla? And so just try to imagine the female chrysops fly will slice through the skin using the scissor-like mandibles and maxilla. So they can actually create incisions on the bite site. So take a look at the blue arrow. It's pointing to a certain area in the skin where there was a previous bite by a chrysops fly. As you can see, there's a small incision. So this video will show us how the mango fly will slice through the skin just to get our blood. So I want you to watch this video. So take a look at this fly. Can you identify what is this? Okay, clue. This is the vector of Onchocerca volvulus, and it is named after its color. Okay, so this is the black fly or the Simullium fly. And this Simullium fly is easily identified by the presence of the hump on its back. So check the hump behind the head. So that's one of the distinguishing features of a black fly aside from its color. You know very well that black fly or Simullium fly is the vector of Onchocerca volvulus. Okay? So during the bite, the Onchocerca volvulus female will introduce the stage 3 larva of Onchocerca volvulus. So, I want you to classify what type of biological vector Simullium fly is in the life cycle of Onchocerca. Is it a propagative, developmental, or cyclopropagative? Female black flies particularly target thin areas of the skin. So that's why you can see a lot of bites on the nape area, the ear, the elbow, and the ankles. Okay? And then I want you to take note that feeding is facilitated by a powerful anticoagulant in the saliva. Aside from the anticoagulant, there's also that chemical that can actually partially numb, okay, the bite site. So that's why it will help the black fly feed on the blood without the host being aware of its presence, thereby extending the fly's feeding time. So again, the black fly tends to bite on thin areas of the skin, the ear, the nape, the elbow, and the ankle. In terms of its mouth parts, I want you to take a look at the three shovel-like appendages. Okay, so these shovel-like appendages are actually sharp, and they are used by the Similium fly to stab the skin of the susceptible human host. And I want you to take note that these mouth parts will also deliver a what? An anticoagulant through the saliva. Okay. So these are examples of bite sites of Similium fly. So I want you to imagine that every time they will bite the skin, they have actually inserted the three shovel-like appendages. Please remember that in the case of Onchocerca volvulus, it is the microfilaria that is migrating. So therefore, what is causing the blindness, the river blindness in Onchocerca is not the adult, but the migrating microfilaria. So the next question is, where can we find the adult stages of Onchocerca volvulus? They are actually found in subcutaneous nodules. So if you will try to biopsy this onchocercoma or subcutaneous nodule, you will be able to find a lot of adults. Okay? And if you're going to examine the eye of the patient, and if you can see a worm, then most likely that is the migrating microfilaria of what? Onchocerca volvulus. And I want you to remember that Onchocerca volvulus can trigger inflammatory processes, and it is also associated with Wolbachia, which can also, what you call this, trigger or further stimulate inflammatory responses. So that's why there is what? Development of blindness. I hope at this point that you can already explain why the disease is named river blindness. It's because black flies or Similium flies are usually found in river water because that is their breeding site. So another picture of river blindness caused by the Onchocerca volvulus. And there is also the development of onchocercoma or subcutaneous nodules containing the adult stage of Onchocerca volvulus. This is just another picture of onchocercoma containing the adult stages of the Onchocerca volvulus. Let's try to identify what is this fly. One of the unique features of this fly is that they have this long proboscis. Okay? Aside from the fact that their abdomen has markings which can also be used to identify them. Ladies and gentlemen, this is the tsetse fly or Glossina fly. Okay? Take a look at the long proboscis and the markings on the abdomen of the fly. Again, appreciate the long proboscis and the markings on the abdomen of the tsetse fly or Glossina fly. So let's try to examine the proboscis of tsetse fly or Glossina fly. The proboscis is actually covered by those two maxillary pulps, the hairy structures that you can find in the picture. Between the two maxillary pulps, okay, you have there the labium and the labrum, okay, that one stick-looking structure there that is composed of the labium and the labrum. And the labium and the labrum are the ones used by the Glossina fly or tsetse fly to pierce through the skin. If the Glossina is not actively feeding, the maxillary pulp will actually cover both the labrum and the labium. But if the Glossina fly or the tsetse fly is actively feeding, the maxillary pulps will separate from the labrum and the labium, and the labrum and the labium now will be inserted through the skin of the human host. So this is a feeding tsetse fly or Glossina fly. As you can see in front of the fly, you have there the presence of the maxillary pulp. And I want you all to take a look at the labium or labrum, okay, that are being inserted into the skin of the human. But if the tsetse fly or Glossina fly is not actively feeding, the maxillary pulp will again cover the labium and labrum. So this video will actually show us a Glossina fly that is about to pierce the human skin, and this video will also show you the separation of the maxillary pulp from the labium and the labrum. I hope you can recall that tsetse fly or Glossina fly is the vector of what? Trypanosoma brucei, right? And I want you to take note that it will introduce through the bite the what? Trypomastigote stage of Trypanosoma brucei. What is bad about the Glucina or tsetse fly is that they can trigger eschar formation at the bite site. So the photo on the left side of the skin is showing us the initial skin lesion, okay, after the patient will be bitten by the Glucina fly. As you can see, there is erythema, okay, and then the patient will be complaining of pain or sometimes of itchiness. And then later on, you will appreciate that the bite site will form an eschar. I hope you can remember that the infection with Trypanosoma brucei will also present with cervical lymphadenopathy, and we will call it as the winter bottom sign. Again, 2 pictures of winter bottom sign. So can you all please identify what fly is this? Yes, this is the vector of the Leishmania species. This is the Plebotomus or sandfly, the vector of Leishmania. Okay, so I want you to appreciate that the head, the thorax, and the abdomen of this fly is fluffy or is covered with hairs. I want you to appreciate that the wings, okay, are always raised above their bodies. And I want you to appreciate that their legs are longer than their bodies. Again, the head, the thorax, the abdomen, and even the legs are covered with hairs, giving the fly the fluffy appearance. Okay? The wings are always raised above the body. That's very obvious in the picture. And take a look at the legs. The 6 legs of the Plebotomus fly, okay, are longer than their bodies. I hope you can remember from the blood and tissue flagellate chapter that the vector of Leishmania species is the Plebotomus sandfly. Okay? And please recall that this fly will introduce the infective stage in the form of what? Promastigote. Okay? And they will introduce the promastigote through their saliva into the bite site. Malophaga, Anoplura, Diptera, Siphonoptera, then we will have the Hemiptera, the bugs. The first medically important are the bed bugs, the one that can actually cause allergic reactions in the skin, right? And the second one is this one. Okay, so I want you to guess what is this. Yeah, you got it correctly. The triatomine bug or the kissing bug. Triatomine, triatoma, kissing bug, reduvid bug. Okay? And they are named kissing bug because they prefer to bite in areas near our mouth. I want you all to take note that this bug is actually transmitting Trypanosoma cruzi, the causative agent of what? Chagas disease. Okay? I forgot to mention that Trypanosoma brucei is actually the causative agent of African sleeping disease, right? Okay. I hope you can remember that triatomine bug will actually introduce trypomastigote, the infective stage of Trypanosoma cruzi. But there's a plot twist, okay? In tsetse fly or Glucina fly and Plebotomus sandfly, they are introducing the infective stages through their saliva. In triatomine bug or triatoma, they will introduce the trypomastigote through their feces and not through their saliva. How is it possible? Take a look at this one. As the triatomine bug is feeding on our blood, it has to empty its intestine to accommodate more volume of blood. And for it to do so, it will defecate. Okay? And I want you to take note, since the bite site will have pruritic reactions, the person will automatically rub or scratch the bite site, right? And what will happen is the person is actually rubbing the feces of the triatomine bug into the bite site, introducing now the trypomastigote. So take a look at this one. This is a triatomine bug that is feeding on our blood. And as it is feeding on our blood, it will actually defecate, releasing now the what? Trypomastigotes. The bite of triatomine bug will actually cause chagoma. Okay? It's a furuncle-like lesion associated with induration and central edema, and sometimes it can be accompanied by lymphadenopathy. And please take note that some of us will also develop allergic reaction to the bite of triatomine bug, and that's why we have here what we call the Romania sign. I hope you can still identify the trypomastigote of Trypanosoma brucei and the Trypanosoma cruzi, because I might use the picture of the trypomastigote and ask you for the scientific name of the insect vector. And that ends the chapter on medical entomology. So this will be the last lecture for parasitology. So good luck sa exam ha. Thank you everyone for listening.